Welcome to ANA Investigates, a monthly podcast featuring the latest in neurology and neuroscience, brought to you by the American Neurological Association. I'm your host, Adeline Goss. This is an exciting moment in the field of neurogenetics. Advances in gene therapy are opening up new avenues to treating certain genetic forms of otherwise fatal and incurable diseases like ALS. This may give genetics a bigger role in the daily clinical practice of many adult neurologists. Today, we'll learn about this frontier in neurogenetics from Dr. Bryan Traynor at the National Institute on Aging. He was interviewed by ANA member and neuromuscular specialist at Johns Hopkins, Dr. Ricardo Roda. Here's Ricardo.
We are happy today to be joined by Dr. Bryan Traynor. He's a senior investigator in the Laboratory of Neurogenetics at the National Institute of Aging. Dr. Traynor is best known for his work aimed at understanding the genetic etiology of ALS. In 2011, he led the international consortium that identified the underlying mutation in a large portion of people with ALS and frontotemporal dementia, a hexanucleotide repeat expansion in the gene C9orf72. His team also identified the same mutation in 1% of patients that clinically diagnosed with Alzheimer's disease. For the purpose of this interview, I'll refer to this as the C9orf mutation. Welcome to ANA Investigates, Bryan.
Thank you, Ricardo. I'm really looking forward to talking with you today.
So, Bryan, what percentage of patients with FTD and ALS have the C9 mutation?
Thank you, Ricardo. That's a great question, and I think it gets to the heart of why there was so much excitement surrounding the discovery of this pathogenic repeat expansion. So it turns out that if you look at familial ALS and frontotemporal dementia, it accounts for between 20 to 30% of familial cases, depending on where you are in the world, which population in the world you're looking at. And then what was even more exciting is that in terms of the sporadic cases of both FTD and ALS, i.e. those that don't have a family history, the expansion accounted for about 6 to 7% of those cases. So overall, talking in terms of ALS, if you add all of those numbers together, there is about 1 in 10 ALS cases will carry this repeat expansion. And I think that it was the frequency of this that made things so exciting for everybody.
What happens to people that have the C9orf mutation? In other words, can you describe to us how this is transmitted and what some of the symptoms of it may be?
I think that your question touches on another important aspect of this particular mutation, namely that it brought two very disparate clinical entities, frontotemporal dementia and ALS, together into one rubric, into one metric. So basically, if you carry the repeat expansion, it is transmitted in an autosomal dominant manner, which means that you only need one copy of it to get disease. And you will either get ALS or frontotemporal dementia or a mixture of those two conditions. So in terms of ALS, obviously we're talking about weakness and spasticity. If we're talking about frontotemporal dementia, generally speaking, we're talking about behavioral aspects to it, although there are reports that language can be affected as well.
Can you describe the strategy your team took to identifying this mutation?
We really used a combination of methods to actually get at the final mutation. First and foremost, it was based on an international collaboration between our group and many other groups around the world who were also looking at this locus. Secondly, it relied on the collection of and phenotyping of families, particularly those in Wales, done by Dr. Hugh Morris, who's a really talented neurologist that was working in Cardiff in those days. And he had spent a lot of time driving down to the little village where all of these patients were and collecting the phenotype and the specimens necessary for our research. And then the third was the development of next generation sequencing technology, which allowed us to sequence across chromosome 9 to a very high depth, and then to analyze that data in a very thorough manner, looking for what might be the underlying cause. And indeed, that was ultimately what gave it to us in terms of trying to understand what the actual underlying mutation is. As an aside, I should mention that we had narrowed the region from about 7 million base pairs, which is about the size of the city of London, because there are 7 million people living in London, on the short arm of chromosome 9. And then using a genome-wide association study in Finland, in the Finnish population, we were actually able to narrow it down to just 200,000 base pairs, which is like a street in London. And in genetics terms, that's very, very small, very, very short. And yet, because of the nature and the complex nature of this particular mutation, the hexanucleotide repeat nature of it, it actually took us 4 years from the identification of that very small stretch of DNA to actually identifying the underlying mutation.
So something that may not be obvious for people that just get a single sentence on what the results of the study is, which is identifying a single gene and a mutation specifically in that gene. How many people would you say are involved or were involved in total in bringing this project about?
So if you look at the paper, there's about 30 or 40 authors, I believe, on the actual paper. The number of authors on this sort of highlight the fact that you need all of these clinicians to go out there and collect all of these samples. And without that, we would not simply be able to do our job as geneticists to actually find the underlying mutations in these samples.
So Bryan, we typically think of genetic disorders as disorders of childhood. However, as we learn more and more, we find out that there are plenty of adult onset genetic disorders.
Yeah, I think that's true, Ricardo. I actually think that if you look across the different fields, that the pediatric neurologists have done a fantastic job bringing genetics into their clinic on an everyday basis. And they apply a variety of genetic approaches in order to diagnose their patients. Over time, that is increasingly becoming so in adult onset cases. And I think it's increasingly recognized that genetics plays a fundamental and central role in the etiology of adult onset diseases, particularly neurodegenerative diseases. It's also interesting that you bring this up because just very recently we discovered mutations in a gene called SPTLC1 as a cause for juvenile ALS. And this is a gene that had previously been described as a cause of hereditary sensory autonomic neuropathy. And now we're extending the phenotype over to juvenile ALS. And I think that it highlights the fluid nature that sometimes exists between adult onset cases and child onset cases. What's particularly exciting about that finding is that it appears that serine, high-dose serine, may be beneficial in those young onset patients and may go some way towards ameliorating their symptoms.
So Bryan, this is an exciting time to be an ALS researcher. It was recently announced that an experimental drug has shown promise in treating ALS caused by another mutation, the SOD1 mutation, which is the first identified mutation that causes ALS. Now, the way this drug works, it's an antisense oligonucleotide, which essentially binds to the RNA and destroys it. So do you think a strategy like this would work for the C9orf?
Yes, I do. So I think that this is an incredibly exciting area to be in at the present time. Remember that SOD1 was first identified as a genetic cause of familial ALS back in 1993. And it's taken us until 2020 that we actually now look that we we have a effective treatment for this particular genetic form of ALS. In contrast, C9orf72 was first described in 2011. And part of the reason why everybody was so excited about this particular discovery and jumped on doing additional research and working on this was because even at that time, it was recognized that antisense oligos were a potential treatment against repeat expansion, this particular type of mutation. And in fact, there is a clinical trial by Ionis ongoing and Biogen ongoing in C9orf72 cases. I don't know what the results are, but I have high hopes that it's actually going to be effective in slowing down or even halting the progression of symptoms in those patients. And I think it would be really exciting for me as a physician scientist who still sees patients devastated with these disorders to go from the discovery of the gene to an actual effective treatment, and I mean effective treatment, in less than 10 years. And I think that that's actually just the pace of moving forward within the field just blows my mind.
At this moment, we don't really have any effective treatments for ALS. However, testing is available. So, Bryan, when should we be testing patients with suspected ALS for C9orf? And if you do have a positive result, when do you test family members, and when is it appropriate to consider testing in asymptomatic family members?
So let me take the first part of that question first and just tell you what I do in my particular clinic. When a patient comes into me with ALS, either familial or sporadic, one of the first genetic tests, if not the first genetic test that I send, unless there's some other clinical indication otherwise, is for C9orf72. And I do that irrespective of the family status, whether they tell me they've got a family history or not. And that's because actually there are more cases of C9orf72 that do not report a family history compared to those cases that actually do report a family history. The other reason why I like to send C9orf72 off initially, not just because it's such a high positive hit rate, but also because the cost of that particular test is on the lower side compared to exome sequencing and Sanger sequencing. And it's only once those C9orf72 tests come back as negative that I then proceed to the second round of testing, which is looking at other genes such as TDP43 and FUS and SOD. I do think that as treatments, and particularly the gene therapy type of treatments, which are very precision medicine based, and as they become available for the different forms of ALS, I think that the paradigm that we see with respect to testing, genetic testing, is going to change. It is going to be a case that we really become much, much more aggressive about genetic testing and getting it done early in the course of the workup of the patient. And of course, the reason for that is because we want to institute such gene therapy treatments as early as possible in the course of the disease. So that's the first part of your question. The second part of your question refers to family members, and these are presumably individuals who do not have symptoms and have no signs of ALS or frontotemporal dementia. Right now, at present, and this is kind of the standard approach that a lot of people, a lot of neurologists, not just in ALS, but also in other neurological diseases adopt. The standard approach is that you send such patients to genetic counselors who will inform them and educate them as to what is involved in testing and how to interpret the results and what this might actually mean for them, either a positive test or a negative test. And that is generally the way it's done, because what we want to do is we want to empower that asymptomatic family relative to make their own decision whether they want the genetic testing. Now, again, I don't think that aspect of testing in asymptomatic individuals is going to change, but what I do think is that the impetus to do that asymptomatic testing is going to change if those precision medicine gene therapy treatments for certain genetic forms of ALS become available. And indeed, this is a topic that has been hotly discussed, widely discussed among neurologists. And I think that we kind of see that we are on the cusp of a golden era of neurology, where we can actually do real good for these patients if we can identify the genetic mutation as early as possible.
That's very interesting, Bryan, and I think it really takes us away from the age of what I like to call nihilism in neurology. So this leads to a different question in terms of thinking of where research is moving forward with ALS and genetics. But should we be focusing our efforts at this time in develop more therapeutics, or should we making a stronger effort into finding novel genes for the other 90% of patients that we haven't identified a genetic cause for?
So thank you, Ricardo. That's a really good question, and it's actually a question that I ask myself increasingly over the last number of years, because the question is, where do we put the focus of my team's effort, and indeed the field's efforts? Should we put it on finding more genes, or should we just say, right, we're going to take those genes that we know today, and we're going to actually just try and make gene therapy, and develop gene therapies for them? I think that the answer to your question is this is actually a mixed approach. We actually use both approaches, the gene therapy and the genetic and the finding of genes, gene discovery, in order to help our patients. Let me explain why that is. First and foremost, I do think that there are genes out there that we can go after in terms of developing gene therapy. And indeed, there are efforts underway to do that, and pretty exciting ones. However, I also think that the cost and the capabilities of genome sequencing technologies is improving in leaps and bounds. So whereas we have not been able to identify the rare causes, genetic causes of sporadic ALS, these new advances in genetic sequencing and indeed in the analysis side of things will allow us to actually identify them. And I think that it would be a shame to miss that opportunity, because, you know, these patients, they're suffering from this disease. And if we're able to turn to them and say, well, look, we think that it's due to this particular mutation, that gives us, again, to use the same metaphor, a toehold that we could use to try and actually go after and to develop gene therapy. I mean, I think it comes back to this notion that you cannot develop gene therapy and you cannot develop a precision medicine approach unless you know the underlying gene. So I think that you're making a distinction between finding genes and gene therapy, and actually, in my mind, they're like a syncytium, they're like a continuum.
Well, Bryan, thank you so much for joining us. I think that the conversation that we had is really interesting. And I think it really highlights, in my mind, a couple of things, which are, number one, arriving at the discovery of one of those genes, it's really a tremendous amount of work, and it takes a relatively long period of time. And so my hope is that in the future, with novel tools and more collaboration, we can really arrive at conclusions a little faster, which mainly I think it's important because discovering these genes is really leading to novel alternatives for our patients, and it can really have a direct effect on their disease. The second point that I wanted to make is that often those that are not familiar with genetics will say, well, what's the point of doing genetic testing? And I think that the answer that we have now is that because it can really lead to therapeutic paradigms, and we can really make a difference for our patients. So thank you so much for highlighting all of these points, Bryan, and thank you so much for joining us. We really enjoyed your conversation.
Well, thank you, Ricardo. It was an honor to be invited to do this, and I really enjoyed our time talking today.
That was Dr. Bryan Traynor, Senior Investigator in the Laboratory of Neurogenetics at the National Institute on Aging, interviewed by Dr. Ricardo Roda, Neuromuscular Specialist at Johns Hopkins. That's it for our show today. You can comment on this episode or pitch an idea for a future ANA Investigates podcast by visiting myana.org and clicking on the Education tab. For ANA Investigates, I'm Adeline Goss.
This has been an episode of ANA Investigates, the official podcast of the American Neurological Association. Thanks for joining us for this episode. If you're not already a member of the ANA, please visit myana.org to learn more about the benefits available to ANA members, including access to an array of educational programs and resources, like online CME courses, webinars, the ANA's journals, the Annals of Neurology and the Annals of Clinical and Translational Neurology, and more. Dr. Adeline Goss is our host. Our producers are Drs. Romer Giucaden, Adeline Goss, Michelle Johansson, Clifton Gooch, Danny Bega, Megan Ritchie, and Jim Sigler. This is your editor, Jen Hurley. Copyright, the American Neurological Association.